Welcome to the Deep Dive. Today we're going to be looking at SCN1A-related epilepsies.
Very cool.
Yeah, we've got an interesting research article from 2024, and it explores how variations in this gene uh, can lead to different types of epilepsy.
Wow.
Yeah, so we'll break it down, and hopefully it'll make sense even if you're not a genetics expert.
Okay, cool.
Yeah, so we're going to compare Dravet syndrome, or DS, with milder GEFS plus epilepsies, and then see how the location of a gene variation can affect its severity, and even uncover the role of something called status epilepticus.
Oh, interesting.
So first things first, can you remind me what SCN1A is? I know it's a gene, but what does it actually do?
Right. So imagine your brain is like a complex electrical circuit, right?
Okay.
SCN1A provides the instructions for building these tiny doorways called sodium channels that control the flow of electrical signals between brain cells.
Okay.
So when there's a variation in this SCN1A gene, those instructions get messed up. And the sodium channels, they don't work properly. This disruption can lead to an imbalance in the brain's electrical activity, making seizures more likely.
That makes sense. So this study focuses on two types of epilepsy linked to SCN1A, Dravet syndrome, and GEFS plus germ. What are the key differences between those two?
So one of the most striking differences is the age of onset.
Okay.
DS usually appears much earlier in life, around 5 or 6 months of age,
Right.
while GEFS plus tends to emerge around 12 months.
Okay.
And DS often starts with these prolonged seizures, especially those triggered by fever.
Oh, wow.
As a child with DS grows, they might experience other types of seizures too, like myoclonic jerk, staring spells, and even seizures that affect their awareness.
So the same gene can lead to these very different types of epilepsy
Right.
with different timelines? That's pretty crazy.
Yeah, it is. And the impact on development can also differ.
Uh,
While DS can sometimes be associated with developmental challenges, GEFS Plus generally doesn't have a negative impact on cognitive development.
so how do scientists figure out whether an SCN1A variation is more likely to cause severe DS or milder GEFS plus MO?
Well, they've developed these incredible computer programs called in silico tools that can actually predict the potential severity of a gene variation. It's like having a genetic crystal ball.
Wow. So they can just analyze the gene and get a sense of how it might affect a person's health.
Exactly.
That's amazing.
And it's not just about predicting severity.
Yeah.
They found that these scores, which have names like Grantham-CAD and Revel, also relate to the age of seizure onset.
Okay.
The higher the score, the earlier the seizures tend to start.
So a high score might mean preparing for seizures sooner.
Yes, that's right. It allows families and doctors to understand what they might be facing and make more informed decisions about care.
Okay, that makes sense. Now, I remember you mentioned that there are different types of SCN1A variations. Can you explain those a bit more?
Sure. So the two main types are protein truncating variants, or PTVs, and missense variants.
Okay.
Think of it this way. If the SCN1A gene is like a recipe for the sodium channels, a PTV is like ripping out a whole page of the recipe. It leads to a complete loss of function of the sodium channel.
Wow, okay.
A missense variant, on the other hand, is like changing a single ingredient in the recipe.
Uh-huh.
It might alter the channel's function, but it doesn't completely destroy it.
So PTVs are the more severe disruptions.
Exactly. And here's where things get even more interesting. The location of these variations within the SCN1A gene also plays a role.
Okay.
The study found that PTVs can pop up anywhere along the gene, but missense variants tend to cluster around specific regions that are crucial for the sodium channel's function.
So those missense variants are targeting the most important parts of the recipe.
Yeah, that's a great way to put it. They're drawn to those functionally crucial areas, which explains why they can still have a significant impact despite being less disruptive than PTVs.
Okay.
And this clustering pattern can give us valuable insights into which parts of the sodium channel are most sensitive to these variations.
It's like figuring out the weak spots in a system.
Yeah.
Knowing these hotspots could help researchers develop more targeted treatments.
You got it. The researchers actually mapped out these hotspots and categorized different regions of the gene based on how many variations were packed into them.
Okay.
They had variant-dense regions, variant-sparse regions, and normal regions. And what they found was fascinating. Those variant-dense regions, which often code for really critical parts of the sodium channel, were strongly associated with DS and an early onset of seizures, usually around 6 months of age.
So more variations packed into a crucial region equals a higher likelihood of DS and earlier seizures.
That's the general trend they observed. On the other hand, those variant sparse regions were more often linked to GEFS plus and a later onset around 10 months.
Location and density really matter when it comes to these gene variations.
Absolutely. Now, this is where the study takes an unexpected turn. It seems that even those PTVs, which we usually think of as causing severe disruptions, can sometimes lead to milder forms of epilepsy.
Wait, really? How is that possible? I thought PTVs were like ripping out a whole page of the recipe.
That's what makes this finding so intriguing. They discovered that these unusual PTVs were mostly found at the very beginning or very end of the SCN1A gene, especially in the last part called the last exon.
So these PTVs are like hiding out at the edges.
Yeah. To explain how they might be causing a milder form of epilepsy, we need to talk about a process called nonsense-mediated decay, or NMD.
Okay, what is NMD?
So think of NMD as your cell's quality control system.
Okay.
It's like having a team of tiny inspectors that check the instructions from your genes before they're used to build proteins.
Okay.
If they spot a major error, like a PTV, they step in and destroy the faulty instructions, preventing the production of a dysfunctional protein. But here's the catch. The research suggests that NMD might not be as efficient at the very beginning or end of a gene.
So it's like the inspectors are more likely to miss those errors if they're at the edges.
Exactly. In those unusual GEFS plus cases with PTVs at the edges of the gene, it's possible that some partially functional protein is still being produced because NMD didn't catch the error. This could explain why they have a milder form of epilepsy.
That's amazing. So it's not just about the type of variation, but also its location and how the cell's quality control system responds to it.
Precisely. It's a complex interplay of factors that determines how these gene variations ultimately affect a person's health.
This is all so fascinating. I'm starting to see how even tiny variations in our genes can have these ripple effects throughout our bodies.
It really highlights the incredible complexity and interconnectedness of our biological systems.
So we've covered a lot of ground already, from the basics of SCN1A to different types of variations and even the cell's quality control system. What else did this study uncover?
Well, they also took a close look at families where multiple members shared the same SCN1A variation. And guess what? They found that the age of seizure onset was much less variable among those with identical variations compared to the overall DS or GEFS plus groups.
So having the same genetic typo makes you more likely to experience seizures on a similar timeline.
Exactly. It suggests a strong link between specific variations and the timeline of symptom onset, which could be incredibly valuable for families and doctors. Knowing that a specific variation tends to lead to seizures around a certain age allows for better preparedness and more informed decisions about treatment.
It's all about understanding the patterns right.
Right.
If you know what to expect, you can plan accordingly. Now, before we move on, is there anything else we should know about these variations and how they affect the type of epilepsy someone might develop?
Yes. Actually, let's talk about status epilepticus.
Okay, what's that?
You know how we've been talking about seizures?
Yeah.
Well, status epilepticus is like a seizure that won't quit. It lasts for more than 30 minutes and needs urgent medical attention.
Yeah.
It's a serious situation that can have long-term consequences.
Yeah. That sounds really scary.
It is. And here's the bombshell. The study found that while only 5% of GEFS plus cases started with status epilepticus, a whopping 33% of DS cases did.
Wow, that's a huge difference. So status epilepticus is much more common in babies with DS.
That's right. This makes status epilepticus a very specific, though not very sensitive, indicator for DS in babies with SCN1A variations.
Oh.
Think of it this way. If a baby with an SCN1A variation has status epilepticus as their first seizure, it's a very strong clue that they might have DS.
So it's a red flag that doctors and families should be aware of.
Exactly. It highlights the importance of recognizing and responding to status epilepticus quickly, especially in babies with known SCN1A variations. Early intervention can make a big difference in managing the condition and potentially minimizing long-term effects.
This has been so insightful. We've covered so much ground already, but I know there's still more to uncover.
You're right. This research raises some important questions about those unusual GEFS plus cases with PTVs. Could further research into those cases help us develop new therapeutic approaches for other SCN1A-related epilepsies? Could we find ways to manipulate NMD, perhaps even restoring some sodium channel function?
That's a fascinating thought. It's like we're on the verge of unlocking a whole new level of understanding about how our genes work and how we can potentially intervene to improve people's lives.
Exactly. And while there's still much to learn, this research gives us hope for the future of personalized medicine, especially for those with SCN1A-related epilepsies.
We've covered a lot in this first part of our deep dive.
Mm.
From the basics of SCN1A to the different types of variations, the role of location and density, the cell's quality control system, and even the significance of status epilepticus.
We've seen how seemingly small genetic changes can have a big impact and how understanding those changes can lead to better care and potentially new treatments.
Join us for part 2, where we'll delve further into the implications of this research and what it means for the future of SCN1A-related epilepsy research and treatment.
It's amazing to think that we're just scratching the surface of understanding how our genes work.
It really is.
Yeah.
And it makes you realize how complex our bodies are and how much we still have to learn.
Right. And this research really underscores the importance of personalized medicine.
Uh-huh.
What works for one person with an SCN1A-related epilepsy might not work for another, even if they have a similar gene variation.
So it's not a one-size-fits-all approach to treatment.
Definitely not. The more we understand about the specific variations, their location, and how they affect the sodium channels, the better we can tailor treatments to each individual's needs.
That makes a lot of sense. It's like figuring out the exact problem with a car before trying to fix it.
Right.
You wouldn't just replace the engine if it's just a flat tire.
Exactly. And that's where research like this comes in. It helps us pinpoint the source of the problem so we can develop more targeted and effective treatments.
So what are some of the potential implications of this research for developing new treatments? I know you mentioned manipulating NMD earlier.
Yeah.
Can you expand on that a bit?
Yeah, sure. Remember how we talked about those unusual GEFS plus cases with PTVs at the edges of the gene?
Right.
Those cases suggest that it might be possible to modulate NMD in a way that allows some functional protein to be produced, even in cases where there's a PTV.
So we could potentially trick the cell's quality control system into being a little less strict,
Yeah,
allowing those partially functional proteins to slip through.
that's one possibility. It's a really exciting area of research because if we could find a way to safely and effectively modulate NMD, it could open up new therapeutic avenues for a wide range of genetic conditions, not just SCN1A-related epilepsies.
That's incredible. So instead of just managing symptoms, we might be able to actually address the underlying genetic cause of these conditions.
Exactly. And it's not just about modulating NMD. This research also gives us a better understanding of the structure and function of sodium channels. By knowing which parts of the channel are most affected by different variations, we can potentially develop drugs that target those specific areas.
So it's like having a more precise map of the sodium channel, which helps us design drugs that can either fix a problem or compensate for it.
You got it. And as we gather more data about the specific effects of different SCN1A variations, we can start to predict who might respond best to which treatment.
So personalized medicine is really at the forefront of this research.
Absolutely. It's about moving away from a one-size-fits-all approach and tailoring treatments to each individual's unique genetic makeup.
That's really exciting.
Yeah.
It feels like we're on the cusp of a new era in medicine, but I imagine there are still challenges ahead.
Of course, while this research is groundbreaking, it's important to remember that we still have a lot to learn.
Right.
We need more research to fully understand the complex interplay between genes and epilepsy and to develop safe and effective treatments based on these findings.
Right. So more research is always needed to kind of connect the dots and translate these findings into real-world solutions.
Exactly, but even with what we know now, this research is already changing the way we think about SCN1A-related epilepsies. It's giving families and doctors valuable information about potential outcomes, and it's paving the way for new and more personalized treatments.
It's a message of hope and a testament to the power of scientific discovery.
Absolutely, and it's important to acknowledge the incredible contributions of the individuals and families who participated in this research. Their willingness to share their experiences and genetic information is essential for advancing our understanding and finding better treatments.
Their participation is making a real difference in the lives of others. So what can our listeners do to stay informed and maybe even contribute to this important research?
Well, first and foremost, stay curious. Keep learning about genetics and epilepsy, and don't be afraid to ask questions. There are many reputable organizations and online resources dedicated to providing accurate and up-to-date information about these conditions.
So knowledge is power.
Absolutely. The more you know, the better equipped you are to advocate for yourself or your loved ones.
And are there ways for people to get involved in research directly?
Definitely, many research studies rely on the participation of individuals with specific conditions, including SCN1A-related epilepsies. You can often find information about ongoing studies through those same organizations or by talking to your doctor. Participating in research can be a valuable way to contribute to scientific progress and potentially benefit from new treatments.
So even if you're not a scientist or a doctor, you can still play a role in advancing our understanding of these conditions.
Exactly. Every contribution counts now. Let's shift gears a bit and dive into a particularly intriguing aspect of this research, those unexpected GEFS plus cases with PTVs.
The genetic rebels hiding out at the edges of the gene.
Precisely, remember how we talked about NMD, the cell's quality control system?
Huh?
Well, the fact that those PTVs at the beginning or end of the gene are sometimes associated with milder epilepsy suggests that NMD might not be as efficient in those regions.
So it's like the quality control inspectors are more likely to miss those errors if they're tucked away at the edges.
That's a good way to visualize it. And this raises a fascinating question. Could we potentially develop new therapies that specifically target NMD?
That sounds like a really innovative approach.
It is. If we could find a way to modulate NMD, perhaps even boosting its activity in those edge regions, it could potentially prevent the production of those dysfunctional proteins, even if there's a PTV.
So it's like fine-tuning the quality control system to be more effective at catching those errors.
Exactly. It's a really exciting area of research with a lot of potential. But as with any new therapy, there are also challenges and ethical considerations to address.
Right. It's important to weigh the potential benefits against any possible risks.
Absolutely. And that's why ongoing research is so crucial. We need to carefully study the effects of modulating NMD to ensure that it's safe and effective before it can be used as a treatment.
So it's a delicate balancing act between pushing the boundaries of scientific discovery and ensuring the safety and well-being of patients.
Exactly. It's a responsibility that we as researchers take very seriously.
Well, this deep dive has certainly opened my eyes to the incredible possibilities of personalized medicine, especially in the field of genetics.
It's an exciting time to be involved in this research, and I'm optimistic about the future.
This has been such a fascinating discussion, and I'm sure our listeners are eager to hear more.
You're right, we've covered a lot of ground, but there's still more to explore in the final part of our deep dive. We'll delve deeper into the potential future of SCN1A-related epilepsy research and treatment, and discuss what you can do to stay informed and get involved.
Stay tuned. Welcome back to the deep dive. We've been exploring this really fascinating world of SCN1A-related epilepsies, and I'm still processing all these insights we've gotten.
It is remarkable how much progress has been made in understanding these conditions, and the pace of discovery, it just keeps accelerating.
It feels like we're really on the verge of a huge shift in how we approach diagnosis, treatment, and even prevention.
I agree. Personalized medicine is transforming healthcare in a big way, and SCN1A-related epilepsies are a prime example of how understanding individual genetic variations can lead to more targeted and effective interventions.
So let's look ahead. What are some of the most promising areas of research that could shape the future of SCN1A-related epilepsy treatment?
Well, we've already touched on a few, like modulating NMD and developing drugs that target specific parts of the sodium channel. But there's another area I want to highlight, gene therapy.
Gene therapy. That sounds almost futuristic.
It does, doesn't it? But it's becoming increasingly realistic. In simple terms, gene therapy involves introducing a healthy copy of a gene into a person's cells to compensate for a faulty gene.
So in the case of SCN1A-related epilepsies, could we potentially replace the faulty SCN1A gene with a healthy one?
That's the ultimate goal. It's still early days for gene therapy in this area, but there are ongoing studies exploring its potential. The challenges lie in delivering the gene safely and effectively to the right cells in the brain.
Right, and I imagine there are also ethical considerations too.
Absolutely. Gene therapy raises important ethical questions about safety, accessibility, and even the potential for unintended consequences. But if we can overcome these challenges, gene therapy holds enormous promise for treating a wide range of genetic conditions, including SCN1A-related epilepsies.
Yeah, it's definitely an area to watch closely. And while researchers are working on these cutting-edge therapies, are there things we can do now to improve the lives of people with SCN1A-related epilepsies?
Absolutely. Early diagnosis and intervention are crucial. Recognizing the signs of these conditions early on allows for prompt treatment, which can make a big difference in managing seizures and improving quality of life.
So education is key. Parents, caregivers, and healthcare providers need to be aware of the different types of SCN1A-related epilepsies and the specific challenges they present.
That's right. And beyond medical management, there's a lot we can do to support individuals and families affected by these conditions. Access to educational resources, support groups, and therapies like physical, occupational, and speech therapy can be invaluable.
Yeah, creating a supportive and understanding environment can make a huge difference. And it's a reminder that while research is essential, it's ultimately about improving the lives of real people facing these challenges.
I couldn't agree more. Behind every genetic variation, every research study, and every potential breakthrough, there are individuals and families navigating the complexities of these conditions. It's their resilience and their unwavering hope that inspire us to keep searching for answers and better treatments.
And it's their participation in research that is driving progress. Their willingness to share their experiences and genetic information is a gift to the scientific community and to future generations.
Absolutely. So, to our listeners, if you're interested in learning more about SCN1A-related epilepsies or getting involved in research, there are many reputable organizations and online resources available. Don't hesitate to reach out and ask questions. Your voice and your participation matter.
This deep dive has been so enlightening. It's given me a new appreciation for the complexities of genetics and the incredible potential of personalized medicine.
It's been a pleasure sharing these insights with you. And remember, even though we've covered a lot of ground, this is just the beginning. The field of SCN1A-related epilepsy research is constantly evolving, and new discoveries are being made all the time.
So stay curious, stay informed, and stay hopeful. The future of SCN1A-related epilepsy treatment is bright, and together we can make a difference.
Thanks for joining us on the deep dive. Until next time, keep exploring.